Okay, so hello everyone. We're going to start the evening session with our second overview talk, and it's my pleasure to introduce Minkyung Baek, who is assistant professor at Seoul University, and previously she did her postdoc with David Baker. She is the lead author of RoseTTAFold, and today she will talk about the past, present, and future of protein structure prediction.
Yeah. Thanks for a kind introduction. So yeah, I'm Minkyung, former postdoc in David Baker's group, and I recently moved to Seoul National University and start my own research group. So yeah, and it's really glad to see you all, and it's great, my honor to give an overview talk in this great conference. So today I will talk about protein structure prediction, the past, the present, and the future. So it's a little bit overselling title. So I will mostly talk about give you just brief overview about how protein structure prediction evolved and what we can do with the RoseTTAFold or AlphaFold with some examples, what I've done or what people in the David Baker's lab done related to the protein structure, protein interaction screening, some protein nucleic acid complex structure modeling, and also designing new functional proteins. So to make everyone on the same page and just give you a brief overview about what will be the previous method for the protein structure prediction. So the best method for the protein structure prediction, like 4 or 6 years ago, is just using some information from the evolution history as well as some template information. So during the evolution, the DNA, our genes somehow mutated, and if that causes some severe changes in their the protein structure or function, then it leads to the disease, and that organism cannot survive. So, or the organism survives through the evolution process, they somehow maintain their essential functions, which means maintain their protein structures. So what happened during the evolution? So if you lose some interactions between residues, that is the key to maintain its structure, then it lose their structure and also lose their function, and that organism just died. And so the organisms that survive through the evolution process, they keep having the essential interactions between the residues and to maintain its structure and their functions. So by basically analyzing the multiple sequence alignment for the proteins having the similar sequences, which is basically evolutionarily related, you can find some structural pattern like these correlated mutation patterns. And from there, you can predict the contact or even distances between residues and generate the protein structures. So that was the coevolution guided modeling, and that was the best method to predict protein structure when you don't have any kind of template informations. So the question is, how can you find this coevolution pattern and from the MSA, which can predict the contact between the residues or even distance and orientation between residues? And there's where AI or deep learning came into play. So deep learning is basically the simple neural network, the hierarchical set of neural network, the layers, which is really good to learn some hidden patterns in the data. So if you provide the system tons of training examples, then these deep learning networks understand all the hidden patterns in the data and predict some useful things or generate something. So people want to apply the deep learning to protein structure modeling. So basically, one thing you can easily So what people first try is apply the deep learning to predict the better contact map or better distance map to get a better structures at the end. So what people formulate that problem is something like this. So you get an MSA and find some covariance features which shows the correlated mutation patterns, and from there try to predict contact, sometimes distances. And you can interpret this process as a kind of AI image processing. And you may want to apply some AI, the deep learning technique widely used for the image or computer vision. So that's why people first apply the residual network to predict the protein structures using some evolutionary history. So this is the early attempt with deep learning. So they first extract some structural patterns from the MSA using some statistical methods like MRF, or just calculating the inverse covariance matrix, or sometimes using simple deep learning network which encode MSAs and to get some structural patterns. And based on that 2D information about the residual interactions, people just use the 2D-dimensional residual network to define contact signals or get a better distance and orientation predictions to make a better risk range to do a folding simulation. And after that, people sometimes apply some classical protein refinement, which try to make the structure a little bit better. So this is the early attempt with the deep learning, which actually happening in CASP13 and some groups in CASP14 as well. And this shows the progress in protein structure prediction with that deep learning method. So using the traditional like template-based modeling or some pre-modeling with the coevolution, it improves a lot, but actually the just applying deep learning, the residual network actually doubled the score. But it's still, there's still a lot of room for improvement, a lot of long way to go to we actually say the protein structure prediction problem is solved. And what happened last CASP, the AlphaFold developed by Google DeepMind does achieve the really good performance, which is very close to our goal, like GDTTS over 90. So at that time, I think almost everyone just so shocked and started to wondering about whether academia actually can develop such kind of good deep learning methods without expert knowledge or without any kind of good computing resource like Google DeepMind. So John Jumper gave a talk at the last CASP conference and just showed this diagram. And I think that's enough to me to get an idea about how to design a deep learning model to predict protein structures. So I found several differences and started to develop the RoseTTAFold based on those findings. So the main differences is the AlphaFold uses some iterative structure feature extraction from the raw MSA. So the deep learning network itself sees the raw MSA, not a processed picture, and try to understand all the patterns in the MSA to generate a better distance prediction between the residues. And also they make it iterative method so that they can actually get all the information from the MSA. And also they generate the protein structures at the end and make it as an end-to-end learning framework so that it actually learns all the features, which is really important to generate good 3D structure. So based on that idea, I generate the RoseTTAFold to see whether academia can make something like AlphaFold without any kind of help from such like Google DeepMind. So we use like 8V100 GPUs to train this model, and I make this algorithm based on the ideas from the AlphaFold 2 method and also some experiment, my own experience using this 3D structures to give another, more feedbacks to get a better structure at the end. So I make this RoseTTAFold, so it's pretty similar to AlphaFold 2. We first try to extract all the structural information from the MSA through some iterative procedure. But the main difference is just generating 3D structure in the middle of the network. And this 3D structure actually gives the feedback to the MSA so that the network can actually see whether current prediction can be realized as a 3D structure, and if not, to correct them. So this is the published last year. And this RoseTTAFold shows quite close performance to the AlphaFold 2. Is still worse than the AlphaFold 2, but I think it's okay-ish because we actually lost in a time, and also we used just, I think, less than 1/10 of the computing resource compared to the Google DeepMind. But anyway, the RoseTTAFold is pretty accurate enough, and we actually can solve a lot of protein structure determination problem, such as molecular replacement in X-ray crystallography. And by using our RoseTTAFold prediction, people can solve their protein structures by using the RoseTTAFold model as a starting point for the molecular replacement. But so that's what happened to me after AlphaFold 2 method. After the John Jumper gave the talk about the AlphaFold 2, and after we make the RoseTTAFold, we want to actually pursue more important problem, which is something like predicting some interactions between proteins, such as protein-protein interaction or protein nucleic acid interaction, and also designing new functional proteins by utilizing the protein structure prediction method. So let me first introduce my work on large-scale in silico DPI screening using the RoseTTAFold and AlphaFold. So when I developed the RoseTTAFold, because I became curious about whether this RoseTTAFold method can predict the complex structure, because RoseTTAFold is based on the co-evolution signal, and if interacting between two proteins are important to do their role, then there should be, you can expect the co-evolution signal between two proteins as to maintain their functions. So because RoseTTAFold and AlphaFold are already trained to extract all the co-evolution patterns in the given MSA. So if you can provide good paired MSA, which is suitable to extract those signals, then RoseTTAFold or AlphaFold itself can predict very good protein complex structures. And based on whether these kind of co-evolution signals are exist or not, you can predict whether two proteins are interact or not. So this is what I first checked, whether RoseTTAFold can predict the protein complex structures of the given already known protein interaction pairs. So these are some examples of the RoseTTAFold prediction. The left one is the ground truth structure, and the right one is the RoseTTAFold model. And you can see a pretty good agreement between them. And also I found that, I think this is the most famous tweet from me about how to use AlphaFold to predict the protein complexes. And I also found that AlphaFold can predict protein complexes pretty well using the same trick I used for the RoseTTAFold. So based on that observation, I checked this second question. We can predict the protein complex structures using RoseTTAFold and AlphaFold by just providing proper paired MSA. And the second question is whether we can predict or screen the protein-protein interactions and using the RoseTTAFold as a kind of screening method to discriminate the protein binders to non-binder. So to screen interactions, we need some kind of metric to decide whether given two proteins are interact or not. So I use the highest contact probability between two chains as an estimation of the interaction probability. So I just summed the histograms less than 8 angstrom and converted it as a kind of contact probability and try to get a highest contact probability in this interchain region and use it as a screening metric. So using that as a metric, we try to benchmark the AlphaFold and RoseTTAFold on PPI screening test. So we generated the benchmark set using the protein-protein pairs experimentally proved in Yeast. And also we mixed that to the randomly generated negative pairs and try to find those real interaction pairs from the negative pairs. So this shows the PR curve of the PPI screening task. So the larger area under the curve means the better performance. So the DCA is the one of the state-of-the-art method, the previous state-of-the-art method using some coevolution signal, and compared to that, RoseTTAFold gives a much better, shows a much better performance to screen the PPI the protein-protein interactions. And we use the RoseTTAFold as the first filter because RoseTTAFold is faster than AlphaFold. And after that, we apply the AlphaFold for the protein-protein pairs, which pass this RoseTTAFold filter. And it gives much better screening performance. So based on that observation, we generate this simple PPI screening pipeline and try to screen all the protein-protein interactions in yeast to see whether this method actually works and find new interesting interactions in the yeast. So this work is done by collaboration with Chen and also other people in the Baker lab. So these models show our predictions for the protein-protein interactions exist in yeast. So it covers a lot of different functions, starting from DNA repair to some enzymatic functions, and among them, the proteins, the complexes colored with blue and brown are the newly detected interactions through this in silico method. And for the others, they have a high throughput experimental evidence, but there's no known structure for that complexes. So among the newly predicted one, we actually do some cowboy biochemistry experiments to see whether they are interact each other and also interact through the predicted interface. So we did a lot of mutation experiments and indirectly prove that they are interact with each other through the predicted interfaces. And we even make the higher order assemblies by manually combining those pairwise interactions. So we made it from trimer to pentamer, higher order complex prediction. And interestingly, one of our prediction, related to one of our prediction, the human GPIT structure was solved and released early this year. And when we compare the complex models, and it looks like pretty similar, and we thought this our prediction probably really close to the ground truth for the yeast proteins. And I'm keep working on improve the RoseTTAFold for better protein structure prediction and also for better protein-protein interaction screening. So I changed the RoseTTAFold architecture a little bit by adopting some good ideas from the AlphaFold 2 paper and also my own experiment. So now it has just pure 3-track, so it always have this 3D track and make an even tighter connection between sequence and structure. And I also introduced the recycling and some model structure to the training set, as well as the complex structures and also the negative PPI sets to make it even better for protein complex modeling as well as PPI screening. So we trained this model, and the RoseTTAFold 2 shows a much better performance compared to the original RoseTTAFold for tertiary structure modeling, the complex modeling, and also the PPI screening. So as you can see, RoseTTAFold 2 is now pretty close to the AlphaFold 2 performance or AlphaFold multiple performance if you use the same MSA. And when we benchmark the PPI screening, RoseTTAFold 2 shows slightly better screening performance. And I think this is because we actually trained the model to do a better screening. So let me change the topic slightly and give you some brief introduction or give you some brief overview about our recent work on extending RoseTTAFold to predict nucleic acid structure and protein nucleic acid interactions. So we basically try to extend the RoseTTAFold 2 architecture to so that it can handle the nucleic acid inputs. So we just added more tokens to describe the nucleic acid. and also added the include the nucleic acid geometry and coordinate to our 2D and 3D track, and modify the sidechain modeling method so that it can actually model the nucleotide orientation as well. So based on this extended version of the RoseTTAFold, we trained this model on the protein complex and protein structures, as well as some nucleic acid structure and protein nucleic acid complexes in the PDB. And this work is mainly done by Frank Di Maio. And with this model, what we named as the RoseTTAFold NA, we can get some promising result on nucleic acid structure prediction. So in terms of the RNA structure prediction, we compare our new method, the RoseTTAFold NA, to one of very traditional method to generate the RNA structures. And as you can see, RoseTTAFold NA outperforms the FAR2 method. But it's not like solving a problem, because it shows a better performance, but it has a pretty low, the aridity as well. So there's still a lot of room for improvement. And when we look into the number of sequences, the correlation between the performance and the number of sequences in MSA, in this case, is still true. We need some sequences in the MSA to model the good RNA structures. And those are some successful examples of the RNA structure prediction use the RoseTTAFold NA method. And as you can see, sometimes with some terminal residues, or sometimes with the residues around the ligand, but overall it looks pretty good, and it finding a good base pairing. And the other important thing, this RoseTTAFold NA actually can predict not only nucleic acid structure, but also their complex structure. So we basically trained the RoseTTAFold NA on protein nucleic acid complexes as well. And as you can see, it knows when it's right or when it's wrong. So the p-aridity is the predicted accuracy estimation from the RoseTTAFold NA method. And this is the real aridity values from the evaluation. And as you can see, there's a good correlation between them. And so you can use this predicted aridity as a kind of metric to decide whether you believe the RoseTTAFold NA prediction or not. And in this case, these are also some successful examples. And as you can see, it actually can predict pretty good DNA or RNA protein structure, protein complex structure, not only the overall trace, but also the exact binding site. So let me introduce my final topic about the protein design. So far, I'm presenting about the protein structure prediction. We try to predict the correct protein structures based on protein sequence and try to understand protein functions based on predicted structure. Protein design problem is opposite problem. You want to design some protein having specific function. So you first think about the protein structures that can have some specific function and try to find sequence that can have the desired structure. So this is pretty closely related to protein structure prediction problem. So Ivan, one of the postdocs in David Baker's group, thought that we can use the protein structure prediction network to design new protein, because it's basically just opposite problem of the protein structure prediction. So we can use protein structure prediction network to predict the 3D structure from the amino acid sequence. So it already understands sequence to structure relationship. So if we use this network to optimize the sequence starting from the random sequence, try to optimize the sequence to have a protein-like structures, then we may end up with new sequence that can have some protein-like structures. So based on this simple idea, he implemented using the Monte Carlo, optimization method. So starting from the random sequence, it gives very blurry distance map, which cannot be realized as a 3D structure. But after a lot of optimization step, it can get a sharper distance map and really good structures at the end. So through the sequence optimization against some objective function, which represent whether the prediction is protein-like or not, it can design new sequences that have some protein structures. And some of them are experimentally validated using the X-ray crystallography or NMRs. But you can use this method to generate any protein, but it's not guaranteed this generative protein actually have some specific function. So the remaining question is whether we can actually input some functional motif or functional constraint to this hallucination process to generate the new proteins having specific, the desired function. So previously we used this free hallucination. The only objective function is protein likeness, but we actually try to incorporate some functional motifs so that the designed protein structure at the end actually have the same functional motif and do a same role as we desired. So we added new objective function so that it can have exact same motif to the given structure, and also we added some problem-specific objective function depending on binder design or enzyme design, something like that. So using this constraint hallucination method, we actually able to generate new proteins that binds to the native target. So we basically try to make a new protein which inhibit the interaction between this P53 and MDM2 protein. So based on this, the motif, based on this interaction motif colored in orange here, we use the constraint hallucination and generate this new protein which colored in gray. And if you compare this motif structure, it's really close to the given motif. And because it has the same interaction motif, when we do experimental validation, this protein actually binds to the target and shows some, and also it works as a competitive inhibitor which is binds to the given protein as we desired. But this method actually takes a lot of time to design a single protein because you basically need to run TR-Rosetta or RoseTTAFold, whatever protein structure prediction network, like 1,000 or over 10,000 times to design just a single protein. So to improve its speed and reduce the computational cost, we want to make a better version or more efficient version to design a new protein. So we reformulate this method, this problem, as a kind of inpainting task, which basically try to complete the protein sequence and structure based on partial information, like the motif sequence and motif structure. So RoseTTAFold already run the sequence to structure relationship. We just transfer on the RoseTTAFold on this inpainting task by providing random partial sequence and structure as an input and try to complete the full sequence and full structure. And by using it, we can actually design another binder which share this interaction motif for the PD1, which binds to the PDR1. So using this inpainting method, we can generate new protein that having this binding motif and also this designed protein also experimentally validated to bind to the target protein. But this inpainting method is basically the predictive model. If you provide the same input, then it always gives the same output. So you can actually cannot get a diverse designs using this inpainting method if you don't introduce any kind of stochasticity, like a turn on the dropout or changing the location of the interaction motif. So we really want to make some kind of generative model for protein design. And there was a really good method, like a diffusion model, what John Jumper introduced this morning. So the people in the Baker lab tries to utilize this diffusion model to design a new protein by changing the RoseTTAFold architecture as a kind of diffusion model like setup. The diffusion model is like you add the Gaussian noise to the given input structure, try to noising it by adding more and more noises, and also train your model to denoise the Gaussian noises so that you can get an original structure at the time 0. So in RoseTTAFold, it already know it trains to generate the good structures based on given sequence, template, and coordinate. So we changes this setup as a diffusion setup. So instead of taking target sequence and some homologous template and some initial coordinate, like a template structures or recycled structure, it gets the currency input sequence and some template, which is the current diffuse coordinate, or some conditions like a target protein, and also the structures as a current diffuse coordinate to the noise. So we just changed the RoseTTAFold slightly, and starting from the pre-trained RoseTTAFold, we trained this diffusion model to this denoising task. And we can utilize this model to various tasks, like using some symmetric oligomer design, or some binder design, or some motif scaffolding by introducing symmetric. So for the symmetric design, what we need to do is just introducing some symmetric noise. And for the binder design, what we can need to do is just initialize the coordinate around the binding interface so that it can denoise around these target interfaces. So using this method, we actually did a lot of work to design a new proteins and also experimentally validate that. So this shows some representative examples. So we actually generate, using this RFdiffusion, we can easily generate the higher order symmetrical assemblies, which was real challenge with the previous RoseTTAFold, the original Rosetta based method. So we tried to generate this C6 or even C8, whatever higher order symmetrics using this diffusion model. So this shows what happened during the diffusion process, actually the denoising process. So starting from the symmetric Gaussian noise, it generates the good C6 structure at the end using this RFdiffusion model. And when we do the EM, we actually can get a very low resolution density map, which our structure, the design structure, fitted well into this low density map. And we also using this method, we also can design a new binders, which has this P53 binding motif. And it actually shows pretty good affinity, which is better than original P53 interaction. And also we can, using this method, scaffolding the symmetric motif, something like the metal coordination motif, and we can generate C4 symmetric proteins, which scaffolding this binding motif pretty easily. And the final thing, I think, is the most coolest thing in this actually paper, which is posted in the bioRxiv today or yesterday. So in this case, we use the RFdiffusion to design a new binder just based on target structure and the interface information. So we basically initialize the Gaussian noise around this binding interface, and using this RFdiffusion model, we generate new binders. And this actually shows really high success rate compared to our traditional Rosetta design method. So with previous Rosetta design method, in most cases, these are the different target proteins, but in most cases shows less than 1% of success rate. But using this RFdiffusion model, we can easily get an initial hit, and success rate is like about 10% or even higher than that. So this is, I think, real breakthrough in the protein design, especially some functional protein design using the deep learning. So this is what my conclusion. So I showed that how protein structure prediction method evolved, and shows the RoseTTAFold and AlphaFold method as a kind of the breakthrough in the protein structure prediction, and we also witnessed even more breakthrough in this CASP. And using this kind of very accurate protein structure prediction model, we can do various things, including just protein or protein nucleic acid complex structure prediction, and as well as the protein design. So the power of the accurate protein structure prediction method is not limited to generate just good protein tertiary structure. It has a high potential, and there's a lot of applications you can think about using that method to do a lot of study related to protein function or generating new proteins. So I'd like to thank all the people involved in this project. And as a new PI, I'm looking for graduate student and postdoc. So if you're interested in developing deep learning methods for to understand protein functions, such as in predicting protein interactions or some mutation effect prediction, and if you are also interested in some developing deep learning method to design proteins, then please contact me through this email. And thanks for your attention.
So thank you very much. Do we have questions?
This is very impressive, Ming. Thank you. Do you have plans on massive release of model structures, like 200 million models?
Oh, like AlphaFold? Well, that's a very good question. So to do that kind of thing, I need a lot of computing resource to generate the models. So, I mean, currently we are using some Microsoft resources and also some Amazon resources, but they are all used to generate another good deep learning model. So if we can get some computing resource for that purpose, then yes. But if not, it would be a little bit hard for me to use our method to generate the huge amount of model structures and make it as a database.
It was really impressive to see your last application where you designed the binder to an epitope. Could you extend this to antibody binding?
Oh, yeah.
Antibody design that binds to a particular epitope.
Oh, yeah, I think so. So this work is mainly done by those people or the processors in this paper. And I'm a kind of more like observer and just give some ideas to them. But I think it's doable. But currently, this is more like a structure generation tool. So it generates a new protein structure that can bind to the target protein, and you need to redesign the sequence to make it actually work. So if you condition this method to generate not only target this binding interface, but also have some antibody-like structures, then it may work as a new designing tool for the antibodies targeting specific antigens. But I think there's still something to consider, like in terms of the sequence design method, so that it can be like having the framework framework has more like antibody structure, antibody proteins, and also yeah, also the interface region has some characteristics for the antibodies. Yeah.
Thank you, Ming. I have a question related to your Yeast interactome. So you basically validated what could be the complexes that are in the Yeast interactome, but probably there are lots of other interactions that are weak interactions. Can you suggest a way of capturing that with your method?
Oh yeah, so it's a little bit hard to say whether it can actually predict those transient or weak interaction using this method, because this is based on the co-evolution signal. So if those weak interaction is important to do their role, then there should be some kind of evolution signal in the paired MSA. But the problem is this method, the RoseTTAFold and AlphaFold, trained on protein structures in the PDB, which usually dominated by the permanent or very strong interactions. So it might it might have weird bias in the training set so that it ignores very weak signals in the paired MSA. So it's a little bit hard to say it can do it can predict the weak interactions, but oh, yeah, it has really high confidence in their predictions. So if you see this PR curve, it has a really high precision but low recall, which means once it's confident about their prediction, usually it's really accurate, but it can miss a lot of interaction which exists there, but it cannot just find out.
Hi, this is perhaps a related question. How do you think the PPI prediction would cope in a case of host pathogen interaction? Because there the pathogen would very much like to maintain the interaction, but the host is trying to escape perhaps. What do you think the remaining covariance information would be strong enough to predict those interactions?
Yeah, that's a very good point. And actually that's the main topic in my research proposal I just submitted 2 days ago. So currently we need the paired MSA to predict the protein, complex structures. And this pairing is based on species information. And we just pair the sequences in the same species with some heuristic method, right? But for host pathogen, they actually came from different species. And pairing should be more like which pathogen target which host. So it's not a trivial problem. And I think it's better to let deep learning or AI just pairing them by using some technique. So yeah, I think that's where we need to do more kind of method development. So currently we are using just heuristic method to generate good MSA. And we also see a lot of engineering effort to utilize a different databases or different MSA generation method. But I think if we can change that to AI or make AI just pair or search the sequences to protein structure prediction, then we may, free from how to utilize or the MSA information from the different databases or such as, or how to pairing them in heuristic method.
Thanks.
Other questions?
This might be a very stupid one, but can you explain the difference in a few sentences between hallucination and the diffusion?
Oh yeah, so hallucination is more like expectation maximization. So it's doing where is it? A lot of sequence optimization directly using some objective function. So you need to define the objective function. So in this case, we actually use the entropy of the histogram so that it can have the more sharper distribution, and also the Ka divergence, the reference histogram, so that it has more protein-like structures. But in the diffusion method, actually there's no objective function during the designing. So it just during the training, it just tried to how to denote the Gaussian noise. So that's starting from the random, so that it can use to generate new structures starting from the random Gaussian noise. So the main difference is that, so it doesn't require any kind of objective function during inference. And the other practical difference is the running time. So using the hallucination to design one binder protein, like this case, it takes more than 10 minutes, but using the hallucination, using the diffusion, it takes less than 3 minutes usually. So it's at least 3 or 4 times faster than hallucination.
Thank you.
Other questions? Okay, so thank you again, Minkyung, and I think we are going to move to the discussion session.
Thank you.